So thank you for coming to Chris' PhD thesis. I can see clearly he has a lot of friends. There's like a number of people presenting in the room. So, so there's two stages of the two steps of the PhD defense. The first step is the open presentation and questions, and then we have a closed session for the committee members for questions and other stuff. Okay, go ahead.
Okay. Yeah.
So, okay. Thank you.
So, hello everyone. Today what we're going to talk about is simulation and control of biological stochasticity. So what I'm going to walk through is three different areas. There's biology, there's mathematics, and there's computation, which all come together to give one story. And what I'm going to start with is the hidden information in biological randomness. So what I want to start by showing you is how mathematics can really reveal something about how randomness inside of biology is used and can be controlled through the mechanisms of the biochemical interactions. So just to start, I just want to make sure everyone's on the same page so that we see that chemical reactions are noisy, right? So if you put a drop of food dye in water, I mean, you say that on average, the food dye, it diffuses and it moves across the water. But it's not some process which is very deterministic, right? This is a process that's generated by all of these dyes. They're bumping into water molecules, and sometimes they go further, sometimes they don't. And so all of this randomness comes into play when you're trying to talk about how chemicals are actually interacting. And somehow, all of biological organisms are random. They're built from all of these random sources. But in the end, they give you something that's deterministic, right? Most animals grow up to be perfectly fine, and somehow that happens even though all of the processes that build it are random. And so I like to give this analogy to be able to introduce that randomness is not something that's just pure random. So when you think about music, right, you can have the same song played in an acoustic rock style or a heavy metal style, where what's different in the signal is the buzz that's going on, that distortion, right? So the same song can have a different amount of distortion, and that randomness is the noise in the signal. But noise isn't just something that noise is on or off, right? You also have something like classic rock, which is right in the middle, right? So you have these different genres, you have these different ideas of the same song can be lots of noise, little noise, medium amount of noise, but you can even hear differences between like amplifiers, right? Everything that has this randomness or this buzz to it has a very distinct signature in its noise. And so if biochemical interactions also have randomness, maybe we can analyze them in the same way that we kind of listen to the noise in music and find something out inside of those chemical reaction networks. So randomness naturally exists inside of the biochemical interactions, and there's two questions that I want to answer. One of them is, biological organisms have evolved to control noise. Like, have they actually, if this noise always exists, and every time that there's some food source in nature, a biological organism will evolve to make use of it. Well, here's randomness. That always exists. Have biological organisms evolved to make use of that? Right? So can they control it, and can they use it? It sounds like that's a very bizarre idea, but what I want to show is that both of these are true. And so all this started with collaboration with the Schilling Lab, where what was shown was that if you take the measurements of retinoic acid in the wild-type zebrafish, you get some measurements kind of like this. This is the concentration of retinoic acid of the wild-type. But if you change this protein CRDP2A, and then you take the same measurements, what you notice is that the amount of randomness inside of your measurements decreases. Right? So, or if you knock it down, it increases. If you add more of this protein, it decreases, without changing the mean level. Right? So the signal of the retinoic acid, of this protein, the amount of it inside the zebrafish stays the same, except its randomness is what changes. So that gives us an idea of maybe there's something to look into here mathematically to see what causes this effect. So can we explain how the mean doesn't change, but the variance or the noise changes? This is what we call mean independent noise control. can we explain why that happens mathematically? So to start, let me give you a little bit of background on what the retinoic acid signaling pathway is. You have retinoic acid, which is a derivative of retinoid, which comes from vitamin A. So this is inside of the zebrafish. It comes from the food source that's there with the embryo. It turns retinoid into retinoic acid, which is then able to freely diffuse around the zebrafish. It can go into the cell, and it can bind to this protein called CRABP. And when it's bound to CRABP, it can go into the nucleus, which can then bind to this receptor, which binds to the DNA. And now the reason why we call this a signal is because this transcription factor, when bound to the DNA, changes the types of proteins that are created. And specifically, this one will upregulate the amount of CYP, and CYP ends up degrading RA. So it's not like a normal degradation in a biochemical pathway, because retinoic acid is actually a small molecule and not a protein. So what it does is it kind of tags something on there, so that way retinoic acid is not able to bind to these materials anymore. And so in some sense, it's like it kind of left the system, so we treat it as though it's, and call it degradation. So this slide gets across the main idea of this whole talk, which is so what these equations are describing is that if you have the retinoic acid, you have a constant rate of chemical coming in, you have a rate which is proportional to the amount of retinoic acid going out. Right? So if you have 2 times the amount of retinoic acid, then you'd expect it to be degrading 2 times as fast. This is just another way of stating mass action laws. But now there's a chance that it turns into RAR, or it goes back from RAR to RA. Right? So they could bind to its receptor, or it could go back to RA. This is a very simplified form of that model. And now we can directly calculate what the mean and the variances are on this, because the model is simple enough. And one thing that we can see, though, is that if this delta equals C gamma, then the mean of this equation doesn't change. And you notice that you have a gamma on top and a delta on bottom, and those will cancel out. And what that means is that if you keep the ratio between the inputs and outputs the same, then you will not be able to change the mean amounts of either the RA or RAR. So the mean amounts of the signal will be unchanged for any change that keeps the ratio between them. And this kind of makes sense, right? So if I say that I double the output and I also double the input, then the mean shouldn't really be changing, because things are moving around faster, but you're not actually pushing chemicals in a certain direction. However, if you have a random fluctuation, let's say you had randomly had too much retinoic acid because the temperature changed, and so downstreams or upstream things changed the amount of RA that was coming in, well, you'd be able to adapt to that fluctuation much easier, just because if the rates are much higher, it's able to push those changes through the system faster. And so as long as you change the rates going down and up by the same amount, keeping the proportions the same, the mean will be unchanged without the variance changing. And that's the core idea. But that has a huge assumption in there, right? This assumption is that delta equals exactly C gamma, where C is some constant. There's exactly some proportionality constant in there, which has to be fixed. And so you might say that's too rigid of an assumption to ever exist in any biological organism. Except we can find a way such that that has to be true. And this is the active intermediate state. So if you now take it one further step, you look at the system with a little bit more detail, what you actually have is retinoic acid binding to CRABP, this binding protein, would be RABP, and then it has to bind to this receptor in order to become the RA bound to the receptor. Now, if you notice that from mass action laws, binding protein, this chemical reaction right here, it requires binding protein, and so from mass action laws, in order for RA to go to RABP, that rate will be proportional to the amount of binding protein that's there. Right? So if you have more binding protein, then more retinoic acid will be able to bind to that protein. But there's also a reverse reaction, right, where the retinoic acid unbinds from its receptor, finds another binding protein, and actually is able to go backwards, right? So every interaction you always can bind and unbind. But this back reaction also requires binding protein, and so therefore that arrow, if you look at the mass action laws, it also has a rate that is proportional to binding protein. So if you ask the question of, is there a proportionality constant which is the same between this side and this side, as though you almost had one giant arrow going down and one giant arrow going up, the answer is yes, that proportionality constant is there, and that is actually the amount of binding protein. So in some sense you could say that this coupling assumption occurs naturally whenever you have an active intermediate state. So what I mean by active is this has to bind to be in the intermediate state. So it binds in the down direction, but also to go back you'd have to bind. So therefore, you have a proportionality constant there, and therefore the amount of binding protein will change the variance in the amount of retinoic acid and RAR. So in both parts of the signal, it will change the amount of it'll decrease the amount of noise without changing the mean amount of protein that's in the signal. So so that's a higher level overview about why you think it's true. We can prove analytically that that's true for actually all reaction rates that you'd be able to choose. And then we can, through numerical simulation, show that what it looks like. So here's what the wild type looks like with just with just some parameter set. And so if you were just to solve it, here's two different solutions where there are two different sets of parameters where you just solve it, and you can see that there's an amount of noise. And when you change the amount of binding protein, you get this orange line from BP knockdown. And you can see that the mean of the solution is the same. Analytically, if you solve it for infinite amount of time, the average of this orange line will be the same as the blue line, except that clearly that signal has a lot more noise to it. So this gives us a way to be able to say, okay, we can control the amount of randomness in biochemical interactions by just having an active intermediate state inside of any of our cascades. So this is all shown on the retinoic acid signaling pathway, but the mathematics of it actually will apply to a lot of other signaling pathways, as long as they have the same property. So okay, that's a very small scale look at it though. This just shows you if you had a single cell with these proteins in it, it's a very confined look at it. You might have the question of, does this actually affect anything in real biology, right? Does it affect a macro scale biological process? Now to get there, we need to start talking about biology in spatial biology. So the idea that people have come up with is called the Morphogen theory, or it's also discussed as the French flag model. So if you have protein that is at one part of the zebrafish or of any biological organism, and you have it in high concentration, then the chemical will diffuse across the animal, right? And so what this is showing is that the amount of morphogen is this concentration, and as you go down the animal, what you'll see is that the concentration will also decrease, right? So as you go further from the source, it diffuses and you get less of it. And the idea is that when you have certain thresholds where if you have low enough of this concentration, then the cell will be able to say, okay, it'll flip what's happening internally in the cell because it will signal it to tell it, I must be over here. So therefore, if I have this high amount of retinoic acid, I will be blue. If I have this high amount of retinoic acid, I will be white. And if we have this high amount of retinoic acid, I'll be red. And as long as nothing has any noise at all and everything's perfect, this model will give you perfect spatial information to every cell, and they'll know exactly where they should be and what cell type they should be. The problem is that reality doesn't work like that. So the reality of positional information, which the Schilling lab, in collaboration, were able to measure this, is that if you actually take a measurement, so like you take the light green points or the purple points, that's just one experimental sample, you'll see that it goes down, up, right? So in space, the amount of chemical that you actually have is noisy. If you go over to this model and you say, well, what if this line was actually jagged? Then what you'd end up saying is, if you had this threshold where if they're actually just reading concentrations, then these cells would get the wrong positional information because the randomness or the noise in the signal would throw them off. And I just want to show this picture again to show you that, here's diffusion in a way that you can see it. And this is not a process which has little bits of noise. It's a process that has tons and tons of noise in it. So how do you get a very noisy process to be something that gives you very precise information, right? Because it's not like your brain forms correctly 1 out of 100,000 times. Your brain forms correctly, the brain forms correctly almost every single time. So how do you get this to be something which is nice like this, that you can actually be interpreted properly? So in the zebrafish, this really matters because what we're looking at is what's called the zebrafish hindbrain. If you kind of take this area in the brain back here, what you have is you have retinoic acid at one part, and then it diffuses down the hindbrain, and you have all these different sections called rhombomeres. And in each rhombomere, you can have cells that form different cell types, and you have different, you actually have distinct boundaries between these, which are sharp boundaries that show up in the zebrafish while it's developing. And so the question is, if we have this very noisy process, this very noisy signal that's telling it where it should be, how come the zebrafish is actually able to make very sharp boundaries between these different parts of the developing brain? And the solution is, well, let's take this local model of noise control and let's apply this to the spatial idea, where now retinoic acid is diffusing around, but every single point you have a cell, and this cell has this noise control mechanism to it. And so we can see this in reverse, where the wild type has the noise control mechanism turned on because it has enough binding protein. But if you knock down the amount of binding protein, then at every single point you have less noise attenuation, and that brings up the amount of noise in the total system. And so therefore you end up with a system that has a lot of randomness in the signal of where it's telling the organism where it should be the red, white, and blue cells. Right? So this is one way of seeing that this local filter, this local noise control, can actually give rise to global features of randomness control and making sure that these processes filter out all of the biochemical randomness that's going on underneath. And there's actually, we can actually simulate the whole hindbrain development, well, just this rhomomere 4 or 5 boundary, where on one end we have cells, I'm not going to go through the entire model, but you have a Hox-Crox model in here, and when they have Hox high, they are one cell type, and when they have Crox high, they have the other cell type. And which one is preferred is dependent on how much retinoic acid there is, with more retinoic acid on this side than over here. And now these are different snapshots going down in time, from 10 hours post-fertilization to 12 hours post-fertilization. And what you see across here is that the stochasticity in the signal plus the stochasticity in the biochemical interactions gives rise to natural boundary sharpening. So you start with something where the cells are kind of mixed, but over time you end up with something where there's a very nice boundary here. The boundary isn't perfect because cell sorting will fix up the last little bit, but you have no cells which are further than one cell part apart. Now, if you knock down binding protein, right? If you up the noise by getting rid of our noise attenuation system, what you notice is that you don't get the same kind of sharpening. And the reason is because remember that background signal which tells the cells where they should be and biases this cell transition model, that no longer is giving accurate information. And so you can have some cells which are way off, like this one over here, that are standing in the wrong area because they're reading the wrong amount of retinoic acid, so it doesn't form the right type of brain cell. And so I just want to highlight here on the bottom that it's actually a very unique property of the system, because if you change most of the other pieces in the system, like here we change the amount of CYP26, if you change other pieces of the model, you not only change the amount of randomness, but you also move where the boundary is. Right? That's why the mean independence is very useful, right? You don't want to make different parts of the brain larger and smaller just to be able to filter out the noise. You want to keep the pieces of the brain the same size while getting sharp boundaries. And so there's really just this binding protein, this mechanism, this mean independent noise control mechanism, is the one that gives rise to the ability to sharpen the boundary without having to move the boundary. But there's another effect that's going on in here. So you might think that, well, if we have this mechanism to attenuate noise, why don't we just get rid of all of the biological noise? Well, there needs to be a little bit of noise, because what people found before was that So if you had the possibility of having two states, so think about these two balls as like they're little stochastic balls with a little bit of random, there's little random wind that's gusting them back and forth. If there's only one well it could fall into, then it would be green. If there's only one well it could fall into, it would be orange. And now if there's two wells that it could fall into, and there was no randomness, no wind that was gusting, then they would just be stuck in one, and it would be there forever, right? And so if they had a chance of starting out green or red, or green or orange, then you'd have this be green or orange mixed in the middle, and there'd be nothing you can do about it. If you have a whole lot of randomness, right, if you have really strong gusts of wind, then the orange ball will go into the wrong spot, and the green ball will go into the wrong spot. And so you'll still have this kind of randomness in the mixing. But if you have a little bit of randomness, then you'll have enough such that the green ball can get gusted out to go to the big well, but this orange ball cannot go to the green one. And so this whole area will become orange. And so what this is showing is that if you have a small propensity to be in one state instead of the other, and you have a little bit of noise, then you'll be able to get a perfect boundary here where it'll be all orange and then green from here on. And so what you don't want is you don't want no randomness, and you don't want a whole lot of randomness, but you want a little bit of randomness in the middle. And we're able to quantify that in a figure where we ran that sharpening picture with a whole bunch of different parameters, and for each of the parameters we found what the effective retinoic acid signal noise is, right? So the effective noise is the noise that we measure after our noise attenuation mechanism, and then we have a certain amount of gene regulatory noise, and we see that if you have too little noise or too much noise, you do not get proper sharpening, which just means that you don't get these cells which are really far off in the wrong area. But if you also have too much noise, or once again too little noise, you also get a problem. And so there's this magical area in the middle, which, if you harken back to one of the first slides, that's your classic rock, right? That's the phenotype in the middle, where it's a special type of amount of noise where the zebrafish hindbrain is able to develop correctly. And this is a phenotype where we're defining it by not too much randomness and not too little randomness, right? So this might give you a whole different way to be able to look at what's going on in a biological organism, because you're not just looking at whether something is happening or happens on average. This is how things are changing randomly at a given time is now affecting whether the sharpening of the zebrafish hindbrain can actually grow correctly. And what we can see is that it's not an answer of yes, no, whether it's high or low. There's actually ranges, a specific range in the middle, which is necessary for these interactions to work. So this is the biology part. The conclusion is that the zebrafish has evolved a mechanism which has an intermediate. And that intermediate is this noise control knob. It's like the distortion knob on an amplifier, where it can, if you give it more binding protein, you get less noise. If you give it less binding protein, you get more noise, and you get that without changing the mean amount of the signal at all. Nothing else about the signal really changes, it's just the amount of noise that changes. But if you have too much noise, of course you can't sharpen because the signal is just not able to be read by the downstream processes. But if you have too little noise, you also can't have this cell switching mechanism happen, and therefore there's this golden area in the middle, this phenotype which is described by a middle amount of noise, which gives you the robust development, the robust hindbrain development. So this is an interesting way to then think about biology, because it says that noise is something that can be both controlled and used by biological organisms. And so before, if you were doing your experiments, you can go, oh, here's some randomness in my experiment. If you do two different experiments in two different ways, if you get different types of noise, that's actually that is a very strong signal about what's going on, and that's not something that you can just discard, because that randomness could actually be a very vital part to how the process is working. So, anyone have any questions on that? That's the bio portion. Yep.
So there's two molecules that are they're like interacting, right? There's more of this one, maybe more of this one, so the ratio is constant. Is there like a time delay? I mean, this one grows and this one grows up, or does it have to be simultaneous? Is it a steady state and it does this, or? Well,
so it's in a quasi-steady state, right? So as the process is evolving, the process of diffusion is still, even though retinoic acid is a small molecule, the process of diffusion is much slower than the actual biochemical interactions of what's going on inside of the cell. And so we take this as the quasi-steady state. And at quasi-steady state, you have these properties of it. Yeah.
Do you think that the amount of noise that is tolerated by cells that are involved?
Yeah. So if you have more cells, less cells, there's another question of how can you know what kind of noise is correct, and how do you fit into the model? How much do the cells actually influence the type of noise in there? That's something that I left out of this, but what we do go into more detail in the paper, and then also in this big thesis right here, is that the way that you can actually look at the noise measurements, and you can back out what the model has to be. So different things like the number of cells will change the noise signals. Different measurements about the noise will tell you whether the noise is exogenous, like it has to be from temperature, or whether it's endogenous, whether it's noise that's produced internally to the system, like the chemical reaction, natural noise. And so what we actually go into more detail, it's kind of a detailed topic, is that every single piece of noise actually has like a signature to it, just like how you hear it in music. And so all those different questions of how much of the noise is dependent on the cells, how much of it is dependent on temperature, and all these kinds of things, that can actually be read by looking at the different noise measurements and different experimental setups. And that's something that I do want to go into more in the future too, but we have the very beginnings of that in that paper. Yeah?
You found in the simulation that there's a sweet spot of both GRN and signal noise where you get proper sharpening. And it's surrounded by a region where you don't get proper sharpening.
Mm-hmm.
Do you have a sense that if you go way out of that area, you might enter a regime where you have proper sharpening again? And if that's just a breakdown of the model, or if that's telling us something?
Well, there's one point. There's exactly one point where...
There's exactly one point.
Yeah, if there's exactly one point at the origin here, well, all the way at the origin, where if you have zero noise in both of them, then you have the theoretical model where everything's deterministic and it'll work out perfectly.
But going the other way?
Going the other way, no, because once you have a little bit of noise, then you'll start to have problems because it can't fix itself. Or you'll have, And in the other direction, everything is just so random that you get nothing out of your simulation. Yeah. So now what I want to talk about is methods for stochastic differential equations. Because what I glossed over was the fact that, I just said, every single one of these dots is a few stochastic partial differential equations which are solved. The stochastic partial differential equation is like 100 by 100 by, so I think these were 100 by 100 by 7 grids of stochastic differential equations. So we're talking about solving millions of stochastic differential equations for each one of these points. And you just do it, and you get this graph. When you talk about that problem, though, solving one stochastic partial differential equation with the standard MATLAB codes that I used, the standard methods that I started with at the start of my PhD, each one of those took about, I think it was like 3 to 4 hours with an optimized GPU accelerated code. And so when I started out, I actually went and told Ching that this was actually completely impossible and I'll never get you this graph. So the next part of this is, how did I get that graph? Right. So really the core to understanding how to make the methods better is to find out what's weird about the biological organism models. And it's something that I end up calling pathway stiffness. So people always talk about, so you have problems that can happen on average. But while we do care about what happens on average in a biological organism, there's very non-average behavior, which is the reason why we're doing the stochastic model, right? And so pathway stiffness, what I'm talking about is these giant bursts that can happen. So here's some x of t, some chemical reaction concentration, and it can either be at 2 or 0. In the deterministic model, if you put it at 0 or put it at 2, then it will just stay there forever. Those are two steady states. But there's enough randomness that it can go off a little bit, and the moment it goes too far, it can flip to the other one. And so if you know your numerical differential equations, you know that whenever you have this very vertical line, you'll have a problem with your numerical integrator that you have to make it so that way your time steps are very small. Now, there are two problems with the way that we were doing these methods before. First of all, they were not adaptive. So if you looked at the literature and you looked at what was implemented in MATLAB and everything, you couldn't find any adaptive integrators, which means you had to choose a step size that you're walking through the simulation such that it would be good for doing these huge changes while also not being too small, so that way you can get through these flat areas that you don't care about. And then the other problem was that the methods that we were using were very unstable. So if this vertical thing ever happened, you'd have to have a very, very, very tiny step size. So when you put those two facts together, you have that very small amounts of time are causing you to have to take time steps which are very tiny for your entire simulation. And so what I mean by tiny is I mean like you're taking steps in, I think they were like picoseconds, in order to solve something up to 5 seconds, all because this little spot right in here just happened to do one change. But those changes are the important part, right? Those changes are the flip between, cell type 1 and cell type 2. That's the stuff that we actually want to get out of our biological models. So we can't just choose a model that gets rid of it. We have to tackle it head on. And we have to find out what kinds of methods can handle large changes that happen transiently. And that's the pathwise stiffness. And so if you've never done Runge-Kutta methods for ODEs, let me just get across the general idea. So the idea is, let's say we're riding a bike on a hill, right? And at any given point, we can know what the slope of the hill is, and that'll tell us how fast we're going. The very simplest method is Euler's method, which says, let's look at how fast I'm going now. So let's calculate, if right now I'm going 10 miles per hour, then where will I be in 1 hour? Well, let's just, if we just assume that we're going at the same speed the whole time, then we'll be 10 miles away in 1 hour. And that's what Euler's method is. Now, Runge-Kutta method is where we just do multiple measurements like that and just add them together in a funny way, such that calculus tells us we get a better answer. So we start by saying, okay, I'm on the top of the hill here, I'm going 10 miles per hour. And that's it. Let's say I want to know how far I'm going to go in 1 hour. Well, instead of going out the one whole hour, let's say if I'm going 10 miles per hour, then in half an hour I'll be 5 miles away. 5 miles away, I'll be, let's say, at the bottom of a hill. I'll be going 2 miles per hour up the hill. This is definitely not those mile per hours aren't good for a bike. You're really slow. But if I'm going 2 miles per hour, I'm walking up the hill, right? And so what you can say now is that, I have one measurement that over here I would be going 10 miles per hour, but over here I'd be going 2 miles per hour. You can use both of those to say, well, what's a better measurement for what my speed will be in half an hour? If you put those two together, then you can get a third measurement that says, in half an hour, I will actually be, somewhere in the middle of the hill. And then you say, well, if I have all three of these measurements, I can say, let's put together all these different measurements of velocity to have a better measurement of where I will be throughout the whole, at the end of an hour. And if you add them together a different way, then you can have another measurement of how fast you'll be at any given time point. And you can see that you can just keep on going and going, right? So the whole process is really just, you take a derivative measurement, you find out what your velocity is, and you use that derivative measurement to tell you how far you might go in some amount in the future to give you another estimate for velocity. You use a previous one to say, well, if I have a better estimate for velocity, let's stick this into the same thing to get another better estimate of velocity. And you keep on pushing this through till in the end you say, well, let's take our, we have 4 different velocity measurements. The magic number is you take 1/6 of this one, 2/3 of this one, or 1/3 of this one, 1/3 of this one, 1/6 of this one, and that gives you some average velocity which somehow makes sense and gives you a very good estimate of where you'll be in 1 hour. So the exact numbers don't actually matter, but what matters is the idea. So the structure of a Runge-Kutta method is you take derivative measurements, and then you use those derivative measurements to give you a better derivative measurement at another spot, and you get a whole bunch of those. You get a whole bunch of derivative measurements where you're just chaining together your previous ideas of how far you'd be given a given velocity. And then at the very end, you use some linear combination here. So you say, here's all my different derivative measurements. You take some weighted average between all of them, and that gives you some miles per hour average throughout that whole step. And then you say, well, we multiply it by how much time it was, you add it to your previous one, and that gives you your new location. So what a Runge-Kutta method at its core is just a table of numbers that say, here's where I want to take my derivative calculations, here's how I want to add together my previous derivative calculations, and at the end, when I have a bunch of derivatives, here's how I sum them up, so that way I get some average velocity throughout a step, and then I use that to get a new point. So in this abstracted sense, you now have a method where, this is actually infinitely many methods, right? It can tell you infinitely many ways where you say, maybe the best way is to, take a quarter step, and then use all those, you can come up with all these different things, and they're all written as different numbers in this vector, different numbers in this matrix, different numbers in this vector, that come together to give you this method of a certain structure. So now we've abstracted our method of, let's come up with, now we've abstracted the idea of let's come up with new numerical methods to, let's just choose numbers that are good. So for stochastic differential equations, you need to add one more spot onto that, right? So we notice that a stochastic differential equation, you have your derivative. So, this is the change in the amount of x is proportional to this is how much you're changing deterministically, and this is how much you're changing on average, or this is how much your randomness is, and this is how much the randomness influences your current change. So this randomness is a Brownian motion term, and we can, and there's good reasons to believe that any of these continuous random processes will have a Brownian motion added to it. But this is the structure of the equation that we want to solve. So it's the same thing that we did before, except now there's randomness that can be had at any spot. So the stochastic Runge-Kutta method is the first generalization that you would think of doing here, which is exactly this. This is what everyone thought to do, right? Basically, it's the same idea, where now what you're doing is you use current velocity measurements and current measurements of randomness to tell you where you should take another measurement of velocity and randomness, and then you just chain those all together. It's the same exact idea, where now we're adding this random term along with all of our other calculations. So if we just had this line, and we just had this line, then we would have the same thing as the Runge-Kutta method. All of these other parts are adding in how much randomness we should have, and these i's are the random numbers. So what a stochastic Runge-Kutta method is just a bunch of time points that we're going to be taking measurements of how fast we're going and how much randomness we have, and then we just chain them together. And if we do this in the right way, we'll end up having a lot less error than if we did it in a naive way. And someone before me named Rossler quantified exactly what needs to be true about those numbers. Right? So I said that this method is defined by a whole table of numbers. If your table of numbers satisfies this, where e is just a vector of ones, so some things like the numbers that you're summing up at the end, the weighted averages, that weighted average has to equal 1. At least the coefficients of it. So there's a bunch of different a bunch of different relations that have to be true about the coefficients, and if these are all true, then you get what's known as a higher order method, which means that each step has a given amount of error, which is less. So we just have to choose numbers that satisfy these. And actually, the guy before me chose a set of numbers that satisfy that. So that's one given group. Where basically what it says is, here's where you are right now. Use that to find out where you'll be 3/4 of an hour from now, and then get a velocity measurement there. And then also take a random number there, and take half of that random number. And now, use your velocity from the past, and 2/3 of your velocity from the 3/4 spot, add the random number, add half the random number, and then there's a funny thing you have to do here. And that somehow, given all the math that's behind it, somehow that gives you a very good estimate of where the random process will be 1 hour in the future. So you might have a question of, I just said, there's all these different numbers. Any set that satisfies these coefficients is a high order method. How do you know that that method's any good, right? So you just chose one set of numbers that works. How do we know that that set of numbers was actually a good set of numbers? So first we have to find out what makes a Runge-Kutta method good. So let's say that on average, we have this movement of our position over time. So this is our time axis, and here's our position. Let's say we basically are going in one direction, where we're going, I'm going across the room, but the way that I'm going is slowing down over time. But if I'm actually moving in a way that, I have a little bit of randomness to it, then your measurement at any given time is probably not what you're going to want it to be, right? So if you think about like a teenager driving in traffic, and you're trying to say, I want to take these derivative measurements and I want to find out where they will be in 1 hour. And let's say you only had one measurement. So you take that measurement and they're in traffic and you happen to take that measurement when they're going 60 miles per hour, right? Because the dude is like driving really fast and then stops. And then you'll say, well, in 1 hour, he will drive 60 miles in traffic. That makes no sense, right? But that's the same thing that will happen in these numerical methods, where if you say, well, how fast are things changing right now? Then I'll use how fast this is changing to be able to say that I will be all the way up here in about half, in one step. That makes no sense. Because in reality what's going on is you have this gentle curve that's going through, and you want to kind of average out this effect of all this waving, but if you're just taking measurements of how things are changing at one given point and extrapolating from it, then you're going to be way off. So this is what's known as numerical stability issues in Runge-Kutta methods. This is a very different treatment of it, but I hope that this is like a very nice visual way to see that any explicit Runge-Kutta method will have a problem, because if you're taking derivative measurements and then just saying, oh, let's just assume that guy's doing the same thing for the next hour, if the process is changing its derivative a lot, if it's changing its velocity, then you're going to have issues because you just extrapolated with a number that makes no sense. And then you'll especially have an issue if they're going 60 miles per hour and then 0 miles per hour, or going backwards. So this ends up being a huge problem whenever you have randomness. And so you can actually quantify whether a method will have problems with this or not. Every method will have problems with it, but some methods will have less of a problem. And so this is defined by its stability region, which basically what you can say is you can take the simplest stochastic differential equation, which is just a process where you are moving at a given rate which is proportional to where you are. So it is saying that as I move across the room, I'm going faster and faster and faster. But it has this random term where as I move across the room, my amount of randomness increases as well. So, and these both increase just by, it's proportional to how far I've gone. So as I go across the room, I'm going faster, but then I get more randomness when I get over to here, right? This is the simplest equation. And on this exact equation, I can tell you exactly how large of a time step a given method will be able to take before it goes unstable. And the reason is because on this type of equation, what will happen is if you take the measurement at the wrong time and you extrapolate too far, then you'll say that it will have gone past the room, right? You'll stay in this equation. You'll say you go below 0. And once I go beyond the room, this model makes no sense. And in this mathematical equation, once you go negative, this will actually exponentially explode. And so you know it's going to go to infinity. So on this very simple equation, you can actually calculate exactly what the maximum step size you can take for any given method is. So that then gives us a way to be able to quantify whether a chosen stochastic differential equation method is any good. When I said that you can calculate it, I meant that in a very theoretical sense, because if you actually write down what the stability region method is, you get this equation where each of these variables are matrices, and then these inverses mean that you're taking the inverse of this, a symbolic equation. And so this actually comes out to, I think it was 3 million lines of Mathematica code, where they are polynomials and 27 coefficients. Each of them can go up to 32nd order. So this is the succinct way of writing down that equation, and as long as you can just find the good numbers that make that equation have a big number, then you're good. The problem, but that's impossible, right? So that's why no one really looked at this problem before, because you can write down what the stability region is, but you can't get any useful information about it because it's just a giant hunk of numbers at that point. Or it's just a giant hunk of symbols. So instead of actually trying to look at it through a mathematician's lens, I decided, well, let's just do this through some kind of an engineering lens. So we have that the method will be stable whenever this function is less than 1. And so what we can do is we can say, well, let's just calculate the whole area where it's less than 1. So if I can put this method onto a computer, I can choose the coefficients for the method, and for that method I can tell you how big of a step size you can take. And so that's really what this is quantifying. This is quantifying, how good is a given method if I chose these coefficients. And now instead of trying to find the best answer by hand, I can just say, here's a number here's a function where I put in numerical methods and it tells me how good it is. Then what I want to do is I just want to find what coefficients give me the best number. Right? I don't want to do this by hand. I want to do this on a computer. So I put this into a global optimizer. Calculating these integrals actually ends up being hard. So I actually got my own supercomputing allocation on the Comet supercomputer and used all their GPUs for about a year and a half. So that way I can just calculate how good, a whole bunch of different methods were for a whole bunch of different coefficient choices. So it's a very brute force method, but what should come out of this are methods which can take really large steps, or at least much larger than the previous methods. And that's what we got out. So what these stability regions are saying is that, if you have, let's say, all the coefficients in that equation were 1, then you can take a step of size of 2. You can take a step of 2 seconds at a time. And the original method, the Euler-Maruyama method that was used before, that would be stable. If you try to take steps of 3 seconds at a time, it would be unstable. It would go to infinity. Now, all the previous methods that Rossler had come up with also had the same stability problem, where they stopped at 2 seconds. So the largest step you could take was 2 seconds at a time. The methods that came out of this optimization procedure were about 10 seconds, right? So which means that while the previous methods were able to step through the equation 2 seconds at a time, this new method is able to step about 10 seconds at a time. And so you can see that, pretty clearly that you're going to be able to solve the equation with a lot less work. There's other features about this where it's able to capture stiffness due to large random numbers better that really make it so that way, in practice, you end up taking about 10 times the size of steps whenever you're going along. And so if you think about it, that should be pretty clear that if you're trying to simulate what happens in an hour, if your previous method was taking steps of around 2 seconds at a time, and the new method is able to step along 20 seconds at a time with the same amount of work, then you'll be solving it much quicker, right? Your numerical solution method, your simulator, will be much more efficient. And so there's one other thing that we had to work out, and that was how do you make this how do you make it adaptive, right? I don't want to take 20-second steps at a time. I want to go, well, there's nothing going on here, so I'm going to take a whole 30 minutes. I'm going to step all the way across 30 minutes, and then when something's happening, I want to take smaller steps. Right? I want to do this adaptively. But there's an issue. The issue is that adaptivity and randomness are two things which don't really combine. And this is something that you can kind of understand in your head, right? So let's say we're going back to one of these car driving examples, and what you want to do is you want to capture the randomness. So you're going along, you're simulating, and there's a huge gust of wind. And that huge gust of wind instantly makes everything change faster. Well, then what you want to do is you want to slow down the simulation. So you want to take a smaller step at that point. So, but what just happened, right? So you're going along your simulation, you take a big step and you say, oh, there's a gust of wind, so I want to pull it back. So what do you do with the randomness, right? So the random event happened in that step, and you want to say, oh, I want to pull back how far I'm stepping. What do you do with that gust of wind, that randomness that happened? If you just say, well, if you just say, well, we took a big step size, let's get rid of that calculation, and let's just do another step. There's a chance that you don't have a gust of wind there, right? So if every single time that a big random event happens, and you say, well, I'm going to get rid of a big step, and I'm just going to go to smaller steps now. If you just do the same random thing that you're doing, you might not get the same randomness. And the issue is that you'll end up throwing out every big random event. If you throw out every big random event, what you'll realize is your simulation will say, hey, big gusts of winds don't happen, and that's why I'm able to simulate it faster now. But that's completely wrong, right? You need to find out some way so that you can throw away steps of your simulation, but still have the fact that, hey, this random process actually happened at a given point, and I need to recover that same randomness, even though I'm throwing away the future. So what's in black is how things are done deterministically, where what you do is you take a step, you measure how much air you had, and if your air was too much, then you would reject that step and use a smaller step. And if your air was too small, then you'd say, that's a good step. If your air was small enough that you say that you're within the accuracy that you wanted with your measurement, You take that, and then you take that step, and you go to your next step. But that naive method does not work with randomness because you'll be throwing out every single big random event. So these blue parts are the parts I had to add to be able to call it the rejection sampling with memory method. One primer before we get there, I said that you have to have an error measurement. The theory is in the thesis and in the paper that says you can get an error measurement with any of these steps. So I don't want to go into it, but for any of these stochastic Runge-Kutta methods, you can have a good measurement of how much error you have in your step. So just assume that I have a good error measurement at any time. So here's an animation now. This is the part that everyone loves. How do you reject steps but keep your randomness and still rebuild the same random path? So let's say this is our position, and we're simulating how the position changes over time of someone that's moving very randomly. And we propose to look at where they will be in the next minute, say. So in order to know how they're changing randomly, we need to take a random number in the computer. So we take this random number according to the normal distribution based off of how the Brownian motion would tell us to. And now we can approximate the solution in 2 points. This is how the error estimate is going to work. And because the difference between those 2 points is the error measurement. Now, if the error was too high, we'd say, okay, we want to reject. We don't want to take that big of a time step. We want to pull it back a bit. So now we propose a smaller step. But we know that this random process, like the amount of wind that's in the background, it needs to get to this level in the future. We can't throw that out. Otherwise, we'll end up throwing out every big random number. So what we can actually do, though, is since this is according to a Brownian motion, if you know one point in the Brownian motion and another point in the Brownian motion, you can actually derive the distribution for what will be in the middle. And that distribution is just right up there. And that's actually the Brownian bridge process. So if you have future randomness and previous randomness, you can actually change the distribution that you're changing randomness such that you'll take the number that will be in the middle. So it's actually the averages of this distribution is right along the line between them, and the variance increases as you get to the middle, and it decreases as you get to the cross of the sides. But what this will do is, basically what it's looking at is if you had a random process where you knew a bunch of different points, and it goes by Brownian motion, this tells you what the Brownian motion would have had, or it tells you a random distribution of what the Brownian motion would have to be in between the points that you already took. And so once you derive that, you can take randomness between randomness, and then you have to make sure you save that value on the stack. So now, this is where the method gets a little bit weird, because now we're bringing in this computational idea of you have a stack. The stack has a piece of randomness on there. And the randomness that we're saving is how far back we went and how much randomness we chopped off. But at the smaller step, now we have an amount of randomness in the step. And so we can say, well, how far did we go in this smaller step? How far did we go in half a minute? And if our simulation then has a small enough error, then we can say, let's accept that step. And then what will the randomness be in this next step if it's this size? Well, it's that value that we saved. And so the key here is that as long as you save your future information and use the Brownian bridge between them, then you can cut back your randomness whenever you want. And if you use a computational stack, you can show that whatever is going to be at the top of the stack will always be the next random point. And so by doing this, you now have you have a random process, but you're able to recreate that random process where you're able to change the step size whenever you want. Now, if you are really following this demonstration, you will have noticed that you couldn't step wherever you wanted. The way that I set this up, you would have had to step exactly this size in the future, because I said that that's how much this random process goes. So you do need to add a little bit to this algorithm. Right. And the full algorithm then looks something like this. So I'm not even going to explain that, but really all that it's doing is it's tagging on one little part on the end, just so that way you can always take a step size of whatever you want. But the idea is what's more important. The idea is just that we can take steps of whatever size we want and still recreate the same random process by using this rejection sampling with memory. So that's why it's called rejection sampling with memory. And it applies to Brownian motion, but it actually applies to any continuous time stochastic Markov process. So any of those, so if you ever do things like tau leaping for Gillespie simulation, there's a way such that you can actually do this on tau leaping as well. That's actually not published, but we'll have that soon. Well, I'm not going to publish it, but we'll put it out there. So what does that mean, right? So I just showed you a few different ways where you get bigger steps, and your steps can change size. How much does it really matter? So here's a model of, there's 20 proteins in there, and in the background, I'm only going to plot just two proteins, so ECAD and VIM. And this actually has this very nice property that this model has 3 different steady states that it would be deterministically. And what you see is that in the stochastic model, it goes between, actually it's 4. There's 4 steady states, but in the stochastic model, it switches between them, and it has that same pathwise stiffness behavior that I described, where when you're at a steady state, it ends up working perfectly fine, and it's a very, very flat process. But whenever you're going between steady states, you end up having these huge shifts. So what you want to do is exactly what the adaptive algorithm does, where it takes step size at around 10 to the minus 4th time steps. So, it takes steps in about a millisecond. But when this giant change happens, it goes all the way down to 10 to the minus 11th steps. So, our average step is about 10 to the 6th, or 10 to the 6th or 10 to the 7th. So about a million times larger than our smallest step. But the fixed time step method will have to take that smallest step every single step of the time. So they have to take at least a million steps more than us. And so the question is, how much of a speed up does that give you? Well, we couldn't actually measure it because it couldn't actually solve the equation in time. So what we ended up doing in the benchmark of the first paper was we said, okay, in this 1 second part, where it won't have these big changes, how faster are we already? And what we're able to show is that against the previous methods, we're at least 60 times faster, 60 times speed up, when you're just doing the constraint problem that has less numerical issues. In the follow-up paper, I added the stability optimized method. So this was done with the Rosler's method with added adaptivity to it. But then if you add our higher stability method that can take larger steps than the Rosler method, then what you have is the SOSRI method, so stability optimized stochastic Runge-Kutta method. This was the Rosler method, where now what we're doing is about 6 times faster than, this was our adaptive Rosler method, so this is paper 1, which beat the previous algorithms by, all the way up to 3,000 times faster. But now we're able to beat that previous one by about 6 times. And so these efficiency gains were actually compounding on each other, and that's how we ended up the whole point of this was to get something that was efficient enough to actually be able to build those graphs, and each efficiency gain, each time you made something 10 times faster, it's 10 times times 10 times and 10 times. It was a process that ended up giving a wild amount by the end of my PhD. And so another equation was the van der Waals benchmarks, and I just want to show this because against previous methods, including previous methods from other packages that exist out there, other software, we're able to show about 6,000 times speed up in the best case. So in the end, what I have was a new method, where this new method allows you to do adaptive time stepping and stochastic Runge-Kutta methods of high order with high stability. And the amount of people in a given room which knows how to all those details is usually 1, right? And that's an issue, right? So I spent all this time to build a very, 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 being able to make sure that you get this right. You can never actually show that it's right in one case because this adaptive time stepping, it has to be on something where you don't have you don't have analytic solutions for most stochastic differential equations. So you don't know how to check your answer, and your answer is only true randomly. So you have to do this for a whole bunch of different times. And so the first thing I ran into when I built all these algorithms was that I had really cool stuff that nobody ever wanted to implement ever again. So what do you do at that point, right? And so that's when I got into software development and this differential equations.jl. So why would I do it? Well, we got this method which is more than 1,000 times faster, but no one's ever going to use it the way that because it is a complicated algorithm. So what we needed to do is we needed to make a software to give it out to people. We wanted to do this for biologists, but the issue was, the first time that I approached Ching with this, he said, oh, it's not going to be useful because there's no way you can simulate biological models. Because biological models, they're not just equations. right? The simplified ones are equations, but the ones that we actually care about in 2018 or 2017, biological models are very dynamic. They change size. You have cells which are going from one cell to two cells, and you have cells which are dying, right? So if you're to look at it in an equation sense, you'd have the number of equations that you're actually trying to solve are also changing. And so if you just write something that solves equations, it's going to be completely useless for the models of 2018. So we have two problems that we have to solve. One is we need to get this out to biologists in a way that they can actually use it. But two, we need to make sure it can handle all the dynamicness of a real biological model. Then it's something that would be useful in this space. So let's look at it like this. Let's say we have a multi-scale cellular model where inside of one cell we have a single ODE or SD, so a single equation. And now what can happen over time is that the single equation can become two equations because the cell splits. We need to be able to solve this kind of problem and just be able to directly call the integrator. So when the modeler says, here's my model, just solve it. And that's what we got. So the 1.0 differential equations.jl, the core of it was to have a new thing called the callback system, such that it can, in 30 lines of code, say, here's my model, here's what I want to solve, use a higher order adaptive specialized method such that you solve this. And what's actually shown here is the amount of x in cell 1, which goes up. So this model, the cell will always divide whenever this protein hits 1. So you'll see that the amount of protein goes to 1 and it divides. The amount of protein goes to 1, it divides. And then it can plot the number of cells, which is increasing over time. So remember that the number of cells is actually the number of equations. So now with this kind of framework where we're using this kind of callback system and the dynamicness in the back end, we can actually encode full biological models of cells and everything. And then the biologist just has to say, there's just this one line in here, like Rosenbrock 2-3, or for the other one, SOSRI. They just say, use your very fancy method to solve these equations that I wrote down. And what it spits out is one simulation of that problem. So we need to go all the way to something which is realistic. So how do you get there? Well, you can encode any model as though it was some kind of pseudo-array. So the way I want to show this is, let's say you have embryos with tissues, with populations, with cells, and each cell has different proteins in there. If you actually traverse this graph and say, this is 1, 2, 3, 4, 5, you can just traverse the graph all the way on the bottom to 20. And then at each point you can say, well, there's also some pieces which are associated with the embryo, right? So something that's in the embryo but not in the cell might be the background retinoic acid concentration gradient, right? So you can have spatial information encoded in the embryo, not with cells. Each cell can have a piece of information. But if you traverse a graph like this, you can just say that, you could put a numbering on every single value that's inside of here. And so what we developed was the program, what it's able to do is it's able to read this structure and able to compile a very specific form of the program, which is specific for this whole entire model. So you can take a full multi-scale model of a biological organism, and then it just knows how to read it, where it says this is equation 1, 2, 3, where the amounts of equations and the pieces between them are able to change at any given point in time. So you can have equations on a given cell, and then you can add or delete cells whenever you want, according to whatever process. So you can say, I have all of these different equations which say how my chemical reactions go over time, and then I have a Gillespie model that says that these cells, they die or were created at a given time. And then when you put it all together, it's something that just works with some added benefits that over time what we've built is all these add-on modules where to any model that you solve, you can have things like you can estimate what your parameters should be from data, you can estimate what your uncertainty is in the model, and all these different things because it was all built in a way such that all of these extra features are just built on top of the solve command, and then this is just some model that you throw into the solve command, and then it all composes from there. So there's a very long feature list that you can actually go through as well. I'm not going to go through all the details because if you want to look at it, look at the thesis, or the best place to look is just look at the documentation online. But the key idea is that we're able to get this flexibility without losing any speed. And the reason is because we're making use of the Julia programming language and its special type system, where every single time someone gives you a type, we don't actually have one compiled program. Every single time someone gives us a model, we compile a specific new model for their program. And so what we're able to do is we're able to show that any given model, we are optimized for that model. And so if you look at the error, so this is the amount of error in log scale versus the amount of time it took to solve the problem, then if you look at, like, say, MATLAB's ODE45 solver or the famous Fortran method, do-3-5, you can see that our new Julia implementations are faster than all the methods that came before it. So, like, if you take a given amount of error, let's say 10 nano, I don't know the measurement, but if you then say, and then you look at how much time it took there, you'd see that it's about 10 times faster than the previous Fortran methods, and that's about 10 times faster than the MATLAB method. So even on the simplest equations, we're able to get about 100 times faster, while having all this dynamicness and the flexibility required to solve biological models. Plus, we have all this stuff for solving stochastic differential equations with all this brand new stuff. So the many researchers over the last, I think now it's been about 2 years, have found a use for this combination of speed and flexibility. So there's a group at University of Michigan which are actually using this for self-driving cars, where this is actually fast enough that way there's an embedded computer in the self-driving car that's able to run the simulation ahead of time, and it's able to use that to be able to adapt what it's doing. So I didn't get there's a full video of this, which matches the point of view of the car along with that. I couldn't get it to work, so I'm very sad about that, but it's really cool looking. Take my word on that. The group at the MIT Robot Locomotion Group is using this on their embedded robots, so that way the robot is able to simulate, it's actually faster than real time now, so it's able to simulate what the robot movements will be like using their model faster than it actually happens in real time, so that way they can use that in the future to maybe come up with some way that it simulates what it's doing and adapts its own actions based on how it looks at the environment. Another group that's using this are the people at the Max Planck Institute for Dynamics and Self-Organization. And so they have all these different pieces, like calculating Poincare graphs to be able to understand chaotic systems. And one way to look at why this really matters is that each one of the dots in the simulation is one solution of an ODE. So they're solving them millions and millions of times, and they're really making use of the extra speed that's required here in order to solve this. And at this point, I just want to show that they even have a live webcast that they did, where in the background what they're doing is they're running this differential equations.jl to be able to, as part of their dynamical systems.jl package, to be able to look at chaotic systems. And then even large quantum physics groups are using it. So one nice feature about this is that since it was all built in a way such that it composes any of the abstractions that you put on there and compiles it to be a fast code, what the quantum physicists have found is that you can put complex numbers in there. You can put imaginary numbers, quaternion numbers, really random numbers I never really thought about. But you put those in there, and it'll compile a specialized code that works for them. And so what these guys have actually been doing is they're using our software with complex numbers and stochastic Schrodinger equations to be able to evolve their models of quantum optics. So it's finding these weird applications far beyond biology at this point. So in conclusion, what I started with was just a talk on biology, right? So I wanted to solve one biological model, which was how do you control the amount of randomness, and how do you find out whether the biological organism is using it? And so there's this really cool result that we get out of it, that there's this mechanism by which an active intermediate state is allowed to control the amount of randomness without changing anything about the mean. And so that was a cool result, but that result took a lot of engineering in the background. And so in order to get there, what we had to do is it was 2 papers of stochastic differential equation methods, one coming up with error estimates and adaptivity, and the other one improving the stability of the methods. And then finally, we built a program that could simulate complex stochastic simulations with relative ease and fast enough in a way that we can solve the model at the very beginning of the talk. So it all came together. There's a reason why I called this simulation and control, of biological randomness, because in one way we wanted to simulate to understand biological organisms, or we wanted to control the noise in biological organisms, but in order to do that we had to spend a lot of time, well, I had to spend a lot of time developing methods to actually be able to simulate those. So it all comes together to one happy ending. And I'd like to acknowledge a whole lot of people. So pretty much everyone in the Me Lab, a lot of people from the Schilling Lab. Thank you for, so the Schilling Lab is the lab that we actually did the zebrafish work with. Tom was here earlier. Tom told me at one point that we were just eating tacos at Taco Rosa that, yeah, the amount of money that it takes to have a student in the lab for a year is about $100,000 or more. And he let me be in that zebrafish lab to learn about how biology works for about 2 years. So like, he didn't have to do that, but he let me in there and I learned all about this biology, and I was able to use this to really put together a very unique view of how biology works, because I had this training, which is not just math, but also the biology, with the help of all the biology and math people here. And then I also have to acknowledge all of my external collaborators, because this is not even half of the people who have been working on differential equations.jl and the Julia Diffie organization. Right, so the software started out as just something for myself to be able to solve this one problem, but now this organization is an organization of 40 people, 7 students a year that are funded each. So Yingbo right here is one of these 7 students that we actually have funded, and it's become its whole own organization that has now grown far beyond my own work. And then the professors of all the previous differential equation methods, I actually got in touch with them through email, and they helped me find the right direction through all this. So thank you, everyone, and I also have to acknowledge, I always forget this one, so the exceed allocation, the supercomputer that I used, they actually gave me a full support guy where I had my own guy that I could contact at any point. And so when I say that I got this working on the supercomputer, I wrote the program and he set it all up on there and gave me the button to run it. So I have to really thank DJ Choi as well. So thank you everyone for coming. Yeah.
Questions?
Yep.
The Runge-Kutta you used, my understanding is explicit now.
Yeah, so what I showed was the explicit ones.
Only explicit.
Yeah, so what's also developed in the second paper is we have implicit Runge-Kutta methods, and we also have switching between them. So we can also find out when a problem is stiff and make it switch between the explicit and implicit ones on the fly. A lot of that is actually with Yingbo. So.
For implicit, you have better stability.
Yeah, well, to an extent. There's the stability of implicit methods on stochastic differential equations is actually something that's a very heated debate. So there's some people in the field that would say that it actually won't work out in the end. Well, we were able to find an L-stable method for only if you had a certain type of noise called additive noise. So if your randomness is not dependent on the system itself, then we have an L-stable method which can take infinite time steps and be fine. Whenever you have a part of the system multiplying the noise, we haven't been able to find an L-stable method. No one else has before either, and there's a lot of talk about whether it's actually impossible. And I could go into more detail on that, but yeah, we are trying to see whether different features like implicitness, or if you know about Runge-Kutta Chebyshev methods, you can tie together a bunch of Runge-Kutta methods just that you grow the stability region. There's a bunch of different methods that we're looking into to be able to see if we can handle the high stiffness, because really these problems are very stiff, and so we want to go beyond beyond explicit methods. But right now, the explicit methods are the best we got. Yep.
Thank you. Very nice talk and nice work. Also talking about the Runge-Kutta method, one reason that it's so widely used by the community is that you can actually prove to what extent it approximates the actual solution.
Yeah.
In stochastic systems, it's trickier because what do you mean by that solution? But you can still do, for example, you can take some kind of gold standard and then compare that to that. It wasn't clear from your talk to what extent you were able to prove that this method, or maybe a similar simplified one, actually does approach the gold standard.
Yeah, so that actually comes from the previous work. So I said that the work by Rosler puts together that theoretical framework for a stochastic Runge-Kutta method, and he proved that given the coefficients. So let me just kind of go back to that. He showed that if you have these coefficients that satisfy these equations, then it will converge with a given order in strong sense. So this is strong convergence, and then these will also converge at order 2 or 3 in weak sense. So as long as you can...
The algorithm applies within the context of this result?
Yeah. So that's why once you've abstracted to here, then I say, okay, any method which has this formulation with these coefficients, we already have the theorem that it will converge. We just need to find out what good coefficients are and make it adaptive.
I see. So even though you're doing a fair amount of additional coding and tweaks, they all fit in the framework of this.
Yeah, it all fits into the framework of this. So it kind of follows like what happened with the ordinary differential equations. Right. So in ordinary differential equations, you first had the Runge-Kutta method. You had RK4. And then a guy named Butcher came along, and he said that a Runge-Kutta method actually follows this whole, this is actually called the Butcher tableau, right here, because he said that, it actually follows this form with this tableau, and then prove that anything which has certain coefficients will then converge in a given way. And then later, after Butcher, you had people like Runge-Kutta Felberg. So a guy named Fulberg came along and said, here's how you do adaptivity. But after that, you actually had someone named Darman and Prince, which had a paper come along and say, well, we can solve this. You don't want to just use any numbers here. The best numbers can be, you have stability of a given size, and they also decrease the amount of error in certain coefficients. So even coming up with the framework of it was actually just the start of ODE theory. The rest of ODE theory said anything in this framework works. Now we just need to find good coefficients. So things like ODE 4.5 is a method from Dorman and Prince, which is an optimized coefficient method, where what their work was just making these coefficients better. And that was one of the impetuses between my method, because I saw that Rossler, this was 2011, I started my PhD at 2013. He just came out with this theory for, this is the structure of a stochastic differential equation method. And then looking at the way that people did it with ODEs, they optimized their methods. This was completely unoptimized, and so I asked the question, how much better could you make it if you actually went through and did optimization here? So that's where my work comes in.
Can you do something like include previous time steps that calculated, gradient at different time steps in order to increase the step size but still retain accuracy, like a momentum idea?
Yeah, so that would be a multi-step method. There have been a few multi-step methods looked at. It's by a couple, Pamela and Kevin Barrage. They're actually a married couple that do math together in Australia now. So I have looked into that. The issue is that it doesn't seem to be improving the stability much, but we are, it's basically they showed that it's possible to come up with a multi-step method but haven't looked too deep into it. Kind of like how here, he came up with the stochastic Runge-Kutta method, showed that one can exist, but didn't go too deep into it. So actually, I'm getting in contact with them. I met them actually in a workshop in London, and I want to see whether this research can go further. Yingbo and I are actually creating an adaptive version of BDF, which will only work for small noise equations. So it has different order conditions if you have noise that's epsilon small. But the large noise case, it just needs more research. We need to do the same thing here where we say, what's required for a multi-step method to converge, and then let's see whether we can find a good one. The work on showing that one can converge has been done. There's just a lot more to do from there. Yeah?
Just a simple question. On your slide on the Watt-Para computation time, did the time for the Julia methods include the compiler time?
No, so I cut the compiler time because it's run 5,000 times.
I see.
Yeah.
Yeah, that's also a question. So if you implement those weight in other language, it will also spin up, right?
So there was actually, yesterday, there was actually a very interesting article on this, that a JIT-compiled language, so Julia is JIT-compiled, a JIT-compiled language can actually make function calls faster than a statically compiled language by C. So someone posted a measurement that Julia can make C calls to shared libraries faster than C can make C calls to shared libraries. And you might ask, why can that happen? Well, it's because these JIT compilers are able to make a lot of assumptions about what they're compiling to, because they're compiling to it at the runtime. And a static compiled library it has. So when you statically compile against when you compile against a shared library, it actually has to have one dynamic step in there, and a JIT compiler can get rid of that. That's one of the things that's going on here. And then another thing is that Julia, if it's all Julia code all the way down, it will actually do a cost assumption and go, well, these function calls are actually small enough that if we got rid of the function call and just copy pasted the code into another one, it would be faster. So there's a lot of, that's called inlining that's happening. So there's inlining that's going on. There's all the dynamic calls are being reduced. There's a lot of engineering that's been gone on into the background to make this faster than, in a lot of cases, what you get with something like C or Fortran, which has then given us the impetus to have this whole organization re-implement all of the ODE methods and all the PDE methods and everything into Julia. And that's what this organization has been doing. And so this is actually just one result. There's a whole repository out there, diffeqbenchmarks.jl, where you can actually go look at all of the different methods and on a bunch of different equations, stiff equations, non-stiff equations, stochastic, non-stochastic, delay differential equations, differential algebraic equations, all sorts of equations. We have this same set of benchmarks, and they all show the same thing. And in a few Stack Overflow posts, I actually quantify how much of this is actually due to different parts of the compilation process and whatnot. So the details are all online, yeah.
What about discrete versus continuous systems? Like you have a system with an integer number of molecules and you want to implement. Does any counterpart exist for such a system?
Yeah, so actually all of these methods work with discrete changes as well. So I didn't want to go into, basically what can happen is we call that a jump problem, and you can take any of these deterministic problems and build a jump problem on top of that. And then what you can do is you can redefine things like the Gillespie method to work where the rates are continuous. Basically, you can say that the Gillespie method, what it's assuming is that a rate is continuous and it solves an integral, but that integral it knows the answer to, and that integral tells it how far to step. And so you can reformulate all of these Gillespie methods in terms of these integrals, and then just add new points to the differential equation on the bottom. And then so you can now solve your model with a few extra ODEs on the bottom, and whenever those ODEs hit 0, that's where you'd have a jump. And so we have it so that we can actually compose all of these ODEs, SDEs, DAEs, DDEs with Gillespie simulations or other discrete simulators. So that actually becomes important because one of the people who is interested in this work, they're actually economists and macroeconomists who look at jump diffusions, where a jump diffusion is described as a stochastic differential equation which has one Poisson process on there. So something like the Merton model in finance. They're these kinds of models, and so we actually solve these using something that ends up it compiles into something that's called a regular jump process, where it's able to find where the jumps will happen, do the jumps exactly, but in between each of them is able to solve the stochastic differential equation. We're also working on regular jump processes, or regular jump solving, where it's like the tau leaping, where you can have tau leaping mixed with an SDE. So we do have that to a very little extent, but making it so that way they all work together and be adaptive, that's one of the next things we're working on. So yeah, we have some of it, but not all of it, but we do have at least more than what's out there, so for now that's good. Yeah.
And one thing that I kind of observed here is that all the Julia methods seem to be kind of converging as error gets very small to about that same time of, what is that, 0.005 seconds? Mm-hmm. For all these different types, are we seeing some sort of limitation on the machine or on the language implementation here? Or is this something that as error would get even smaller, we would see some of these methods which are performing better, some worse than that?
Well, this is a phenomenon. So when the rate at which you converge it's important when you get to lower and lower error, right? So what happens is higher order methods do, or lower order methods do better when you want less error, and higher order methods do better when you want lower error. What I'm only showing is up to 7th order methods. So the methods which you actually want to be using over in this regime, the 8th order method, 9th order method, and we also have these things called the Fegan 10th order, 12th order, 14th order method, and then arbitrary order methods you can also come up with. Those are what you want to use in this regime, such that you wouldn't get this behavior.
So this is just a coincidence based on what you looked at in the region that you...
Yeah, well, this is just a coincidence based on the methods that I chose to build this graph. Yeah.
Okay, so let's thank Chris again.